Right, so welcome to lesson 5 of your ecology unit of work. So as you'll see, lesson 5 is all about field investigations and how some of the ecology work you've been talking about in the last couple of lessons has been put into practical application. So to begin with, pause the video, you review your previous notes, review any extra resources you've got, and have a go at the knowledge check questions, and then we will go through these momentarily in a moment when you unpause the video. Right, you've had to go through these knowledge check questions, so mark them, correct them, change them as you see fit. Define an ecosystem to begin with. If you remember, it is the interactions between living and non-living parts of an environment. What are abiotic and biotic factors? They are non-living and living factors in an environment that affect populations of species, population numbers. NOS dot for short, numbers. What is a food chain? Food chains show feeding relationships between animals in an environment. What is a population? It is the number of a species in an environment. So what is a quadrat and how is it used? Remember, it is a metal frame, which is often a square, and it's used to estimate population numbers in a specific environment and populations of non-moving organisms. What's the transect and how is it used? It's a line along which you use a quadrat, so it's the second use of quadrat, to measure the spread or the distribution of a population of organisms, again non-moving, such as plants. Why is quantitative data important? It's numerical, so we can make sure it's accurate, and it can be statistically analysed easily. And then from all your sort of science experiments, what's the term independent variable mean in the context of an experiment? It is the thing that you change in an experiment. Now, obviously, this lesson's a little different because it just is covering one of the required practical bits of content. Now, obviously, if you are watching this and you're not able to be working in school on this, it's not something you'll be able to do practically, but if we can understand the method and get the method across in this, that's the most important thing. So, as it says, the overview is measure the population size of a common species in a habitat using sampling techniques to investigate the effect of a factor on the distribution of this species. So this is the purpose of the required practical that you can get exam questions on. Where it says measure the population size, so when we're talking about that, we're talking about the number of a species in a habitat. Habitat, if you remember, is where it lives. So using sampling techniques to investigate the effect of a factor, so this could be abiotic or biotic, on the distribution of this species. So we're talking about the spread of location of the species. Where is it located? Where is it densely populated? Where it might be more sparse. So the summary, it says, in this practical, if you're doing this practically, you will work in a group to use a quadrat. So if you remember, our quadrat is usually our 1 meter squared metal frame, to estimate the size of the population. Remember, estimate is what we might refer to as a scientific guess, where we don't know the exact amount, but we can do procedures and steps to try and improve our accuracy as to the number that we're talking about, and the population size. So we're talking about the number of this plant species in a survey area. And remember, they only sample plant species because plants are non-moving, not like animals. Also, you'll use a transect line and a quadrat to investigate the effect of light intensity. So remember, light intensity is an abiotic factor because it's not living, on the number of plants in a survey area. Now, if it's a transect line, you would use it to measure the spread. And the quadrat used to estimate the number. Now, this is the important bit. If you are reviewing this having done this in class, it's absolutely fine because you'll have seen the practical application of it. If not, though, can I ask you now to pause the video, and in the resources you've got with you, go and find and watch this YouTube clip for me, please, and make a note down of the method steps and also any variables that are involved in this practical, because you'd be expected to know the method steps. You might want to do it on the student's notes page at the back of this lesson. Remember, when we're talking about variables, we're talking about the independent, the dependent, and control variables. And as we've said earlier, the independent thing is the thing in the experiment you are changing. The dependent variable is the thing in the experiment you're measuring. And the control variables are the things that you are keeping the same. And this is for fairness and accuracy and reliability of results. So, if you've not done the practical before, make sure you pause the clip now, go and watch this video and make a note of the method steps. All of them, step by step, so you've got that understanding and that visual representation of what the steps to this practical are. Once you've done that, come back to this, unpause it, and we'll go through them ourselves in a little bit more depth. Right, you should have unpaused the video. So, flick on to the next page. Now, this is the point where you would be, if you're in class, working with quadrats and doing this outside yourself. As we're not, and as we're utilising YouTube to get this across, we're just going to sort of talk about steps in the method instead. So, if you remember, the point of using a quadrat is to take an area such as this, place your quadrat at various points, count the number of species in each quadrat, and then use that count to estimate the total number of species in the total area. Now, as it says here, collect two numbers, one from each bag. Use the numbers and tape measures to locate the first position of your quadrat. Lay the quadrat on the ground. What this is getting you to try and remember is that your sample area, before you do anything else with it, you would, with rope, tape, markers, you turn it into a grid, and a grid that has coordinates. Because by selecting numbers, you could then take those numbers and create a coordinate which would show you where to put the quadrat down. So you'd locate it using prepared coordinates. Once you've done that, count the number of plant species in your quadrat, and you would repeat these steps several times so that you would put the quadrat at, let's say, 10 different locations. You would count the species in it at each. Then, to work out your estimated population of plant species from your area, you would then be taking, now instead, this isn't very well presented, so scribble that out and change this to estimated population size equals the average species per quadrat times the total sample area. Both of these would be in meters squared. Oh, well, this would be meters squared. So that would be the way in which you'd then use that data you'd collected to calculate your total estimated population species size. Now, if you were doing a transect, what you're doing here is you are looking at an ecosystem. So let's say you're looking at the plant species on the floor and how they grow in proximity to a tree. If you cannot tell, that is a tree. Now, you might have a couple of plants there, and then you might have a few more plants there, and then the further away from the tree you might get more and more and more and more. That would be a tree, and these are plant species. Now, if you're using a quadrat, what you do is you place down a ruler, tape measure, whatever, and you would simply put the quadrat at different intervals along the tape measure, and you would count species in the quadrat at intervals along the tape measure, or the line, so the tape measure put out is our transect line. So for example, your intervals might be every 5 centimeters like they're on the table, and from there you'd be able to see a spread of your plants. Do they grow not a lot in one location? Do they grow a bit more in another location? Do they grow more in a different location? So if you looked at this very crude diagram, you'd say that the plants grew more the further away from the tree, and would prove that by counting more of them in that location compared to that. And from a biological point of view, we'd know that that's the case because they're further away from the roots of the tree, so they're not competing for light and they're not competing for water as much. So it gives you an idea of how we'd use transect for the quadrat for a transect line, and how you would use it for, as you've got a method up here, estimation of population size. So you've got two examples there, and that's what that video goes through in quite a bit of detail. So hopefully you've got your head around that. Now, if you've had a look at that, turn over, and then you'll then see on your next page, there is a question in which you can have a go at applying some of this information. And it's best probably if we go through this together, because it gives you a bit more idea of how to go about using this idea of quadrats and transect lines. So a student wanted to, to begin with, if you want to have a go at it independently, pause the video, have a crack, and then start the video again. And if you want to walk through the answer with me now, just keep watching. So, checkpoint. A student wanted to measure how distance from a water source affected the size of a plant. Write a method to carry out this investigation. Now, distance. That's a key word here. Distance is not population number. So this is telling you that we want the method for a transect line. Method for a transect line. Now, if it said a student wanted to measure the number, or the student wanted to estimate the number of plants, that might imply using the quadrat to estimate the sample. But this is telling you to use a transect line. So we're trying to look at the spread. We're looking at the spread of plants from a water source. So the spread of plants near or far from water. That's what this question is asking us to do. So if we think about the first step, it's transect lines, so we need to put out a tape measure. So the first step would be lay out a tape across the area. Now, where would it start? A student wanted to measure how distance from a water source. Lay out tape across the area, from the area starting at the water source. Second thing you want to do is, as we said, the quadrat needs to be placed at specific intervals. You should then decide to place the quadrat at set specific intervals along the line. So, e.g., every 1 meter squared. Every 1 meter squared. So, then what you need to do is place the quadrat. So, place the quadrat as above and record. Now, what are we recording? The number or the size? And record the size of the plants in the quadrat. And then obviously you repeat all the way down the line. You repeat at set intervals all the way down the transect line. Now, what would be the final steps? The final steps would be obviously looking at your data and comparing. Compare plant sizes recorded to each distance interval to see if size changes. So give you an idea of how to use that methodology. Right, good. So if you then turn over, what you'll see is, having looked at some of the methods of using a quadrat, the remainder of this lesson, because obviously you're not doing practical applications is to be having a look at applying it in some exam context. So we'll go through a couple of these questions, then I'm going to leave you to have a look at these questions yourself, and you can communicate with your teacher accordingly about the answers. But if we model through a couple of these and give you an idea of how to apply what we've talked about to some exam question context. So the question says, a student was asked to estimate how many clover plants there are in the school field. The image below shows the equipment, quadrat, tape, identification key. Throw a quadrat over your shoulder, count the number of clover plants inside the quadrat, repeat step 1 and 2 4 more times, estimate the number of clover plants in the whole field. What is the tape in the image above used for in this investigation? So the tape is used for distance, isn't it? So in that respect, what we need to be using the tape for is to measure the area of the field. So we can come up with an area of the field that we can multiply our samples, our sample numbers from each of the quadrats by to get our estimated population number. Now, the teacher told the student that throwing over the quadrat over the shoulder was not random. The method could be improved to make sure the quadrats were placed randomly. Suggest one change the student could make to ensure that the quadrats were placed randomly. And if we remember back to previous lessons, we spoke about using a random number generator that you can use to make coordinates which give you a place to put quadrat in the field, which has been sectioned into a grid. So for example, you might have your field, and before you've generated your coordinates, you've turned it into a grid, and you've given each section a number. So you might have put 1, 2, 3, 4, and then you might have put A and B. And from your random generator, you might have generated a coordinate of A1. And in that case, you put your quadrat there. That's what we mean by this methodology. Then, on to the remaining part of this question. How could the student improve the investigation so that a valid estimate can be made? Weigh the clover plants, compare the results with another student's results, count the leaves of the clover plants, place more quadrats, place the quadrats in a line. Now, if you remember back to the question, the investigation itself is asking you to estimate the number of clover plants. So which one of these is going to get a more accurate estimate of the number? Weight is going to have nothing to do with it. Comparing the results with another student's results. No. Count the leaves. We're talking about the plants, not the leaves. Now in a line, that's distribution. That's not number. So that's going to give us about the spread of the plants. But if we place more quadrats, that's going to get a more valid estimate because we can increase accuracy of our average clover per quadrat calculation. D. The table below shows the student's results, number of clover plants counted, total 40, so been added all up and done 5 quadrats. The area of the school field was 500 meters squared. The quadrat used in the table above had an area of 0.25 meters squared. Calculate the estimated number of clover plants in the school field. So, if we think about these, we've got 40 plants. We've got 5 quadrats. 40 divided by 5 equals 8 plants per quadrat. So that means we've got 8 plants per 0.25 meters squared. We've got 8 plants per 0.25 meters squared. And our total area of the field is 500 meters. So, if you just bear with me 30 seconds, we need to find out 0.25 meters squared goes into 500 meters squared. How many times? Now, 500 Now, if we have 0.25, we want to turn it into, we want to know how many times it goes into 500. Divide it. 500 divided by 0.25 equals 2,000. So now we want to go, we've got 2,000, 0.25 meters squared in 500 meters squared, and per each of those we've got 8 plants. So we want to find out how many plants as an estimate is in this 500 meters squared. So we now take that 2,000, we times it by 8. So 2 times 8, as you know, is 16. So our estimate would be 16,000 plants. Sorry, it wasn't written down there or on screen. So hopefully you now see the methodology. So how many plants per quadrat? 40 divided by 5 gives you 8 plants. Each quadrat is worth 0.25 meters squared. So you've got 8 plants per 0.25 meters squared. 0.25 meters squared goes into 500 2,000 times. So you then need to times that 8 by 2,000 to give you an estimate of 16,000 plants. Right. Giving you a good example there of how to do one of these questions. Now, what I want you to do is... Oh, I've just realised we've got the remainder of this question. Not finished it. Apologies. What was the mode result in the table? Mode. Go back to your maths teachers. Tell them it's most frequent. So which one of these popped up most frequently in the table? You've got 1, 2 11s. Now, suggest which quadrat could have been placed under the shade of a large tree. Give one reason. Now, we're looking for plants. Do plants grow well under large trees? I would say no. So our quadrat would be 5. Now, we said it before earlier in the lesson. If you've got a plant under a tree, are they going to find it easier or harder to get water and light? They're going to find it harder. And so our reason is it's harder for them to compete for water and light needed for photosynthesis. Right, you've got a couple of good examples there. So now it's your responsibility and your focus to look onto question 2, question 3, and to try and put these into practice using some of the ideas we've talked about. Now, your teacher might communicate with you a little bit more and tell you want to do all the questions, some of the questions, but they're there for you to have a focus on now and to communicate with your teacher as to whether you've got stuck on any of question 2, 3, 4 or 5 or whether you are happy with them. So we're not going to go through all of these, else our video would be about 6 hours long, but have a focus on them now, and then we will go through the end of the knowledge check questions of this just to finish off looking through this. But the key thing is question 2 onwards, talk with your teacher and see which of these they want you to focus on. And then to finish, if you've gone through those questions and looked at those yourself, final knowledge check on this lesson, state how to make the sampling random at a transect. As we've said, a random number generator. And what are they going to create? They are going to create coordinates to place your transect, your quadrat at. Habitat is where an organism lives. The difference between mean, median, and mode. Mean is your average. Median is the middle number in a lined up data set. The mode is the most frequent number that occurs. So how to estimate population size? So remember, average number per quadrat times the total sample area. Then explain why you take multiple readings of quadrats. What does it do to your estimate? It makes your estimate more accurate. Now, suggest where you would use a transect to take data rather than a quadrat. Not to measure the number or estimate the number, but to measure species distribution. Don't worry about that last question. It's not very well phrased. And then state 3 control variables when using a quadrat for sampling. Using a quadrat for sampling. Now, when you sample, the time of day is important. Trying to keep that the same. The species, so obviously keeping the same species that you're counting, and then the actual quadrat, the piece of equipment, is the same. So the area of which you are counting each time is the same. Good. So slightly different lesson there for lesson 5, and I appreciate that it's slightly picky considering it's a practical that you can't quite do yourself if you're working remotely, but it should give you a good overview of how to use a quadrat to estimate population size or to measure the spread distribution of a population over a specific environment.